Swayam Prabha. Digital India. Educated India.
Hello. Let us now discuss how we can classify the different tissues and organs depending upon the complexity present in that. Like if we see the different tissues and organs in our body, then they have different level of complexity. Like starting from skin to kidney or heart or pancreas or even trachea, esophagus, so different tissues, they have different types of complexity. In some certain cases, the tissue is they don't have that much simple, relatively simpler in complexity, and also they are like two-dimensional structures. Mostly it is the two-dimensional structures, flat tissue structures, and but in certain cases the organs are very complex, like they have a very typical macrostructure, microstructure, and the they have different compartments. Their the microstructure is very different. So all these things. So we'll discuss some of these aspects, like how we can classify them according to the different level of complexity, so that when we devise strategies for bioprinting of that particular tissue construct, we can be like, suppose for low level of complex tissue, we can use device one kind of strategy, but for very high complex tissues, our strategy has to be again very different, so that we can replicate that kind of complex structure, so that we can generate a functional organ tissue or organ structure. So basically this all these different tissue, based upon their macro structural complexity, they can be classified into four different types, like low, starting from low to high. Then the very first thing which will come, the flat tissues will fall into the lower level or simpler level of complexity, such as skin and other tissues. Those are mostly flat tissue structures. Here they have relatively simpler structure. Then the next level of is the tubular structures, something like blood vessels, airway tracts, all these things, where it is mostly, it is also the complexity is not very high, but they have a typical macros. The macro structure is like a hollow structure, sorry, not a tubule like structures. Then the next level of complexity present in the hollow structures, where such as bladder, urinary bladder, and other some other organ, where their their complexity is little higher than the tubular structures, because they have a typical microstructure is not only it like a tubule but the hollow, but they have a typical architecture, or because of that their function is also very different. Then the if you see the highest level of complexity present in the all the solid organs, like kidney, heart, liver, pancreas, all these different organs, they have very high level of complexity, because their structural details, they have very high level of structural details. So we need to understand all these things, and accordingly we need to devise our strategy to target any of these organs for bioprinting. And accordingly, if you see in the literature, you will find the same thing, like people generally started with bioprinting of simpler complex structures like tissue, skin, and all this. So where the the strategy is also very straightforward, and so that the depending upon the technical capabilities of the bioprinter, where it is much easier to print flat structure than a more complex structures, like kidney or liver. A flat structure, the printing flat structure would be much more easier thing to do. So that's what, if you see tissue and organ engineering community, they started with this simpler simpler complex organs or tissues, and there to do that, if suppose if you are going for from small low to high level of complexity, then accordingly also structural requirement also increased with the increase of level of complexity. That means enhanced structural requirements are needed for repair of tissue or organ of higher structural order. So that is also we need to consider. Suppose for bioprinting of skin, what is required for bioprinting of skin would be different than what is required for bioprinting of kidney or liver. So because kidney or liver, they have a very complex structure, so that's why their structure requirement will be very different than the skin, because skin is mostly a flat tissue. So we can start with the 2D array bioprinting, we can definitely we can print them, and we can actually come up with the skin tissue structures. The other important thing to remember here is, as the complexity increases from one level to the other level, the metabolism requirement, metabolic function, so the metabolic requirement also increase tremendously. So that's what also we need to consider. So increased metabolic functions are required, and that has to be supported within the structure in the bypass. So that's what, suppose the metabolic requirement of a liver and metabolic requirement of a skin is very very different. Liver is highly the function highly uh, means liver is a function that is very complex, and that their metabolic requirement is very very high, right? So that's what we need to understand, and we need to also uh, consider during bioprinting of this thing, when you are trying to bioprint a particular tissue structure with the this kind of different complexity. So all these are actually, if you can if you can understand the complexity, and if you can devise your strategy accordingly, then probably you can come up with better strategy, better planning, a better strategy for development of that, or bioprinting and development of a particular tissue or organ structure. Let us start to understand each of these things in some detail, so that when you are trying to, suppose when you are trying to bioprint a particular, suppose you are trying to bioprint a flat tissue or a tubular structure or a hollow structure or solid organs. So what are the things that has to be remembered? You should remember on how you can devise. So there are some guiding principles or rules that actually we can discuss, and that can help you to accomplish any of your bioprinting task. Flat tissues, they are mostly composed of cell sheets stacked in multiple layers. You can think of some flat tissues like the cell sheets. They are arranged in a layer by layer manner, stacked, so that you can, so flat tissue can become, think of like that. And accordingly, so in bioprinting, you can also print like this, different cellular layers and layers of cellular cells. You can print, then you can come up with the whole tissue. So skin is one example of that flat tissue, and it is the largest organ in the body, because if you see everywhere, most of the body is covered with the skin. That's because skin provides you that physical and immunological barrier, so that it can be protected from the outside environment, right? So that is the main function of the skin. So, and that's why the skin, if you have that's why there is a enormous requirement of strategies, enormous requirement of skin for different, to address the like they different, so to address the problem of different skin wound healing, or due to trauma or injury or burn, where the skin loss is the primary, skin loss is huge, maybe can be huge. So there we need to devise strategy how we can print a skin, and we can put that for better tissue regeneration, so that the first better and faster tissue regeneration. And because of that simplicity of that tissue, it is because it is not very complex. So it was one of the fast tissue engineering targets for biofabrication community or even tissue engineering community where the first started working on this skin tissue engineering or bioprinting of skins because of its relatively structural simplicity, and that because it provides skin, if you can divide the skin into, it can be crudely divided into two to three layers, like the topmost is the epidermal layer, then the below that there is the dermal layer, and then there can be hypodermis and other layer. So but dermis and epidermis are the most considerate layers that is required to be bioprinted, and therefore because of this, for providing a barrier function, so that we can, there is a skin loss, then we can put them there, that can be, there can be placed or implanted so that it can prevent, again it can prevent the inside tissue from the outside environment or from this thing, or acts as a barrier membrane. So so now suppose for printing the skin we need to different like this, yeah, the dermis where the fibrous cells and some vascular tissue is also present. The fibrous cells are the major component of this dermis layer, and the epidermis keratin cells are the major cell present in the epidermis layer. So accordingly, we can print them in this fashion to bioprint the whole skin tissue. Other than this, also other important thing to remember here is the for flat tissue, because they are not the depth is not very high because they are mostly thin structure, so the nutrient diffusion within the structure, from suppose from the below surface, the nutrient diffusion to the structure, that is cells present within this suppose bioprinted tissue, because of this thin nature, that can be the nutrients, oxygen that can easily diffuse to the tissue, and there is not a stringent requirement of a complete vascularization, because without actually getting the whole tissue vascularized, we can basically supply nutrients, oxygen, all other metabolic requirements also that can be addressed without vascularizing the whole bioprinted skin tissue. So that's why this, because that's why this bioprinted skin, even without any vascularization, they can sustain and they can be useful, and they can basically they can perform the function. So that is one thing. But vascularization, but again, vascularization is important, and some amount of vascularization is very much required to be have. But that's a not very highly stringent requirement for flat tissues like skin. But rather than very highly complex and thick tissue like maybe liver or kidney, where vascularization is a very highly required to, otherwise the tissue cannot be, the organ cannot be sustained. Another important thing is over the last 2 decades, researchers have been working on this skin tissue bioprinting, and if you see in literature, most of the structural elements of the skin are reproducible through bioprinting. That means people have or researchers have already worked on bioprinting of different skin grafts, skin transplants, like where partial or full thickness skin grafts have been bioprinted, and then have been used. So there are, and they have different preclinical studies, even animal studies also done to, or even some in certain cases the preclinical studies are also done with this bioprinted skin graft, and those were really very powerful, and they can help to regenerate that particular defect or uh, diseased skin for, and will be helping the regeneration of that part. So that's why so that is mostly achieved, but the challenging part, that is still remaining, people have been like a few of the a number of researchers have been working on developing hair follicles that is present on the skin, and also the glands, different glands, also sebaceous glands, sweat glands, those are present in the skin. So these kind of specialized structures, these are also very much important for the function of the particular skin. But suppose if there are no hair follicles, or if there are no glands present in your bioprinted skin, so that skin cannot perform the functions, right? So that's why those are the challenging thing, and there are various researchers who have been working on this to address these issues. But the most of now the most of the research focused on developing this specialized structures on the skin, like hair follicles, glands, and also other things like how to generate nerve bendings, incorporate or integrate nervous tissue within the skin graft, so that the bioprinted skin can have the sense, the tactile response can be possible. So those things actually are the challenges, or those things are the current focused area where people have been working. But mostly skin, there are a lot of research, a lot of research work has have been done on this thing, on skin tissue bioprinting, and you can actually go through different literatures to understand more and more on this. Then there are certain uh, tabular structures, like when the tubular sorry not tubular structures, like tract, like airway tract or blood vessels. Those are like tubule in the form of tubules, like when they are also form composed of cell sheets, but they are formed into circular bilayer structures, bi to trilayer structures, where the cell sheets are arranged in a particular order to get these circular tube structures. Like blood vessels are a natural example of these tubular structures. Even the airway tract or food pipe, like esophagus, those are also tubular structures. So what happens where, like, the in the blood vessels, in case of blood vessels, the innermost cell layer, that is the endothelial cells, they form the innermost layers, right? And they also maintain a functional barrier between the thing that is flowing through the tubes to the through the other structure, right? So that they also present a functional barrier. So there is a tight junction between the endothelial cells is very much important, where they, because of that, the functional barrier that exists. Right. Then the on top of that, so the innermost layer, and then on top of that, a smooth muscle and connective tissue form the outer layer, right? So that provides support, stability of that innermost structure, so that the whole blood vessels can be developed. Right? Now here also, so now accordingly, so suppose if you are when you are targeting bioprinting, then accordingly you have to develop this innermost layer structures and the outermost connective tissue and the smooth muscle and connective tissue layers that is present on the outer layer. So that these two, so these two bilayer tissue structures you have to develop, and that also in it in the tubular format, the tubular shape you have to maintain, that so that this micro and macro structure that you have to generate when you are targeting when you are developing strategies for bioprinting of the tubular tissue structures. Now here also there is a requirement of nutrient deficient and oxygen exchange, but that requirement is also not very stringent because of this thin wall thickness. So the nutrients, oxygen can diffuse to this suppose this bioprinted tissue structures if the complete vascular network does not develop at the beginning. Then also the bioprinted tissue or organ can sustain due to the oxygen diffusion from the sun, from the surrounding area, or from the probably from the the culture from the media. And but with time, that blood vessels or the vascular network needs to be developed for proper supplying nutrients and oxygens to the bioprinted chain. Other than this, if you see, there are large number of work have been done on the development of large diameter blood vessels. But large diameter blood vessels are actually, they are relatively easy to fabricate, because of the dimension is such that with our current technological capabilities, like the bioprinter that we have, with that we can able to generate, or we can able to fabricate large diameter blood vessels very easily. So where, because these are biliar tissue structures and thinner biliar tissue structures, as I have already mentioned that they are blood vessels. So and you can maintain the different that cellular arrangement where the innermost is endothelial, outermost are the smooth muscle cells or some kind of or other supportive cell types like fibroblasts and other cells you can use, and then you can print the whole blood vessel structures. That is it. But by printing of smaller dimensions in vascular or blood vessels like arterioles, capillaries, venules. So these things, when as the dimensions go lower and lower, so that with the lower dimensions, so those things are really typically challenging to produce. And most of the researchers now are they are targeting to bioprint those small blood vessels. So that is because they are also very important and they can work and the tissue level, certain cases at the tissue level, or so they supply blood to the in more uh, deep tissues, so where it is very much required. So these are the challenges lying there. And typically current focus is also to develop or fabricate these kind of structures with bioprinting. The next is the hollow structures. Hollow structures typically they are formed. They consist of the epithelial cells that is present in the inner layer of the thing and surrounded by smooth muscle forming an outer layer. So there can be connective tissue present within this, or some in certain cases connective tissue is not present. But typically this is the structure how they are thing. So that is the macro and microstructure of that hollow structures. Urinary bladder is a very great example of this hollow organ, but there are certain other organs also that people have been targeting to to develop bioprinting. But here the metabolic requirement is typically higher than the earlier two, like than the flat and tubular structures. Typically in this case, because the function of this the tissues is typically yeah, energy demanding. So that's why the high metabolic requirements is very high, and they have certain functional parameters, and also they have there's a complex interaction with other organs. Because of all these things, fabricating this kind of tissue, this kind of organs with hollow structure organs is challenging, and a greater amount of study as the method. You have to devise a very good strategy to work on this kind of hollow structures. But there have been successful regeneration of bladder tissue we using autologously derived urothelial and smooth muscle cells. So this kind of work actually have been done. There is a successful story here where uh, Dr. Anthony Atala from Wake Forest Institute of Regenerative Medicine, they have, and so Dr. Anthony Atala and his group, they have developed device strategies by which they have generated artificial urinary bladder, and that then seeded that with patients or autologous in urothelial smooth muscle cells, and that was implanted to the patients, and those patients doing the patients are doing perfectly fine. So that's a successful regeneration of bladder tissue, and where macro and mesostructure elements of hollow structure organs can be replicated. So when you are trying to do these things, do this generate these hollow structures with bioprinting, then this macro and mesostructure elements of this hollow organs can have to be replicated, so that so that the your organ can function the way it is required to be. But to recreate that organ microstructures, including the glands and vascularity that is present within that particular organ, is highly challenging. And to do that, that's a that's a difficult task. But of course, that would be the most rewarding also, because and designing or devising strategy to do that is the current focus of bioprinting community. So there are various researchers working to accomplish these tasks. So the next level of complexity present in solid organs, or the solid organs, where that means the complexity is the highest order, like kidney, liver, heart, pancreas. These are most complex organs, and it's very difficult to bioprint them with our current technology and with our current knowledge, because we have not reached a stage where we can actually can print a complex, fully functional complex organs like solid organs. So there are major hurdles due to the developmental process and tissue complexity of these organs, because these organs, they are highly, there is a very difficult to reproduce them. And these organs, because they have very high metabolic requirement, so that their demand, like this, their oxygen demand and other things is also very high. So we need mature blood vascular network, so that the, and all with extensive branching that is required, so that the cells, suppose if bioprint that kind of thick structure, anything bioprinted structure, flat thinner structure is fine. We need not to we need not to provide a vascular network. But if thick structure cannot survive without presence of the vascular network, so we need to provide vascular network, because this has been seen that in the it has been a few people, researchers, they have also proved that the cells, they cannot survive if they are present more than 200 to 300 micron away from a blood vessel supply, from a supply of blood vessels, like the blood vascular supply. So that kind of thing. So that means you need extensive branching that is crisscrossing the whole bioprinted construct, so that the each and every cells present within that thick construct get nutrition and get metabolically active, and then they function. Otherwise they cannot sustain. So that is very important for things. So natural vascular network with extensive branching is a highly required, is required for these things. Other this thing, precise organization of multiple cell types is also required, that where, like, so other than this is in the solid organs, if you see, the cells are organized in a perfect manner. That means that organization is very important, organizing that. So if we want to try to recreate that kind of structure, then we also need to come up with that particular organization, that precise organization we have to mimic to achieve the function of that particular tissue. So current focus of this bioprinting community is to develop biomaterial based approaches, and also to use 3D bioprinting, the different 3D bioprinting methods to come up of these different solid organs like this. There's various researchers working on this to generate artificial kidney, liver, heart, pancreas, because these organs are in high demand, and there are end stage failures of this type of organs, where there are many various patients who are actually basically waiting for this kind of transplants, and so that many patients can be saved if we can develop this kind of artificial tissues and organs. Now we'll see how we can actually come up with this kind of different organ tissues organs. Now we'll break them into different things. Like, first we'll see the cell sources, from where you will get the cells for to accomplish this task. Now if you see, mostly when if you are working on bioprinting or biofabrication, or suppose if you are trying to print a particular organ, then you need cells. And if you see the cellular concentration in that particular organ or tissue, it is enormous. Like, there are, in certain cases, anywhere between 10 to 100 million cells are present in a centimeter of, or in a ml of tissue, right? So that much high cell number is, so cell number is huge number of cells are required to bioprint any of this tissue structure. So typically in the bio ink, That's what in the biobank we maintain a cell concentration of anything between 10 to 100 or maybe more than that, 10 to the 6 or 10 to the 6 to 8 number of cells per ml of biobank that is required. So that many cells are required. And if you are targeting for transplantation, then you have to use a cell source that is not immunogenic to the patients. That means that will not cause any immunogenic reactions or it will not be rejected by the body. So there are some potential sources. Stem cells has definitely be one of the most potent potential source for source for bioprinting, because it can be differentiated into particular cell types, and actually we can collect these stem cells from the patient body itself. We can either like bone marrow stem cells or adipose derived stem cells, we can easily isolate from the patient cells, and we can basically grow them and then we can use them for bioprinting. So so there are different types of cell source like human embryonic stem cells are one of the things, but they are potentially controversial, and we have seen that like there have been some stem cell banking where people collect the stem cells from the earlier embryo, and then that has been preserved. Either it can be from umbilical cord tissue, umbilical cord derived, or anything that is basically preserved, and that can, so with the hope that it can be used in future for that particular person. So but that is controversial. There are also different resident stem cells that has been isolated from different organs from the from the patient's body or from a person, and then that has been preserved, that can be used for this thing. There are also some circulated bone marrow derived adult stem cells present. There are a number of researchers have been working on that, where they have devising strategies how we can actually isolate these bone marrow derived stem cells, and that can be used for this thing. iPSCs is another candidate. iPSC basically induced pluripotent stem cells, and you know very well these are induced cells, like from there basically these are from other primary cells that have been again reversed to the stemness. Their stemness is reversed, so that they have been now in the pluripotent state, and then these iPSCs can also again differentiated. So these are basically de-differentiated cells from primary to de-differentiated cells, and then from there again it can be differentiated to different other cell types. So that is, so these are the primary sources of different cells that can be used for bioprinting. But if you are not directly using working for transplantation, then you can use any other different other cell types, cell lines available, and some primary cells also available. Those also you can use for this thing. So far more than 100, or maybe this number is every day this number is increasing, more than different 100 different types of cells have been tested for bioprinting. They have been used for bioprinting applications. That means some researchers, they have used these cells for bioprinting applications or biofabrication, when they have passed, either they have used extrusion based bioprinting, inkjet, or other bioprinting. Like these are some of these things, some of the cell types, but this is like only a few I have listed here, but there are many more than that that can be used, that have been used, and that can be used. But most important thing is no significant difference in viability of all these cells compared to manually plated, because for cell culture, the cell culture process also, there's manual process where we typically we syringe out the cells, and we can plate the cells. That process also there is some date the viability also. But when you do bioprinting, the viability is not very different. More than 90% cells are viable within this bioprinting process. So that's suggesting that the cell printing technique can be applied to most of the cell types. So what are the different cell types present? Most of the cell types can be used. This bioprinting can be used for bioprinting of different cell types without compromising the cell viability. And if you see how to bioprint, there are requirements like in 3D bioprinting, basically that's a 2D array bioprinting. We have discussed this before, like because it's a layer by layer manufacturing technique, so that there we put the cells in one layer, then simultaneously layers are printed, and then we count things. So there are various requirements in that case, in that domain. Like we need either, in case of extrusion based bioprinting, the viscosity requirement is such. We have already discussed this in our earlier classes. Please refer to those lectures. and you will be understanding in great detail what is the role of viscosity for bioprinting, all these things. So we need some pneumatically or some pressure driven, mechanical pressure driven uh, valves, like extrusion mechanisms by which we can basically we extrude these materials, all these things. And also we also need the temperature control for the dispenser. We can control the mechanic the rheological properties of the bio-ink, the mechanical properties of the bio-ink, and so that, and also the physical state of the bio-ink, so that we can use, as I have mentioned in the earlier lectures, we can control the size, like the shear thinning property. The viscosity can be modulated, even the gelation can be modulated, all these things can be modulated, and the temperature can be a factor that can help in doing that. Also, we need to devise strategies by we can do the simultaneous printing of different biomaterial cell types to come up with a suppose in particular in typical different tissues or organs, different cell types present, and their cell organization also very different from one to another. So all these things can be done by simultaneous printing of different cell types within these things, and a wide range of hydrogel material that is required for bioprinting, and also that the age typical gelation mechanism or cross-linking mechanism is required to do that. So all these things are very much required to print a to bioprintable types. And then there are different hybrid printing technologies required, where now suppose uh, as I mentioned, one one type of bioprinting technology cannot be used for printing very complex structures. So probably we need to combine different technologies together. So the hybrid printing technology can be the way to go forward. Other than this also, there are certain requirements for 3D bioprinting organs, like we need tissue specific bioinks, and that should be bioactive as well, so that support that particular tissue specific cell growth. Suppose when you're working with liver, so I need to come up with I need to develop a bioink that is specific for this liver tissue development. So the liver cells, like hepatocytes, they grow very well within this thing. They grow well, they do their functions, all these things will be very good. Another thing is the tissue mimicking nano or micro architecture is very important, as we have already discussed in this lecture, that each tissue or organ, they have a typical nano to micro architecture. We have to also develop this kind of architecture along with bioprinting. And different cell types and extracellular matrix components, probably we need to use them depending upon the tissue type. What are the different cell types, the resident cell types present in that tissue, that we have to see, and accordingly we have to use what are the different extracellular matrix component of the tissue, what is the composition, we need to understand, then we need to replicate these things. Other than this, also living cells can be patterned within this hydrogel tissue scaffolds, and many a times that is very much important, where we can actually basically pattern the cells within the scaffold, so that the organization or arrangement of the cells in that particular tissue, in that tissue types, organ types is possible. Other than, probably in certain cases, we need to also include certain bioactive factors or growth factors that drive the tissue formation or the organ development. And in that respect, spatiotemporal delivery kinetics is very important, because this growth factor delivery is also again another another thing another important thing to design design or device, where we can control the delivery of these factors as per the requirement and of these things. Other than this, the vascular networks, as I have been telling this from the very first day, that vascular networks are the driving factor of the tissue. So all this growth of the tissue. So we need to design and develop vascular growth factors accordingly, so that we can come up with strategies for developing the a functional tissue construct, so that can sustain, that can be viable, and that can grow well. So all these things are, please remember on all these points when actually, if you are taking any bioprinting assignments, or if you are interested to the bioprinting, then please remember this point. These are very useful for to do any bioprinting jobs, and to again also to develop a strategy, or to design a bioprinting strategy or actually to go for organ printing. Thank you very much again for attending this lecture. We'll see you in the next lecture. And thank you very much, all of you.